Okay, I got a little chatty, and there's one more section of the notes on sex-linked diseases. So I'm sorry, we've got 2 videos to watch. But this is actually really a huge concept, the idea of a heterozygote advantage. And so later in the year when we talk about evolution and gene pools, and wouldn't this gene eventually get out of the gene pool if people are dying from sickle cell anemia? It's this idea that it's kind of hidden there, and not only is it hidden, it actually gives an advantage. So we'll use this example again when we get to the evolution unit. Here's a pedigree, and so they're actually going ahead and showing you who is heterozygous. Here they're calling it a carrier. It's a little tricky. It is co-dominant, but they call it a carrier because I don't see it in your physical body. I can see it when I look at your blood example, but I can't see it in your body. So the last part is on sex-linked traits, and there are 3 that you probably want to go ahead and try to memorize because they're so very common. The first is red-green color blindness, the second is Duchenne muscular dystrophy, and the third is hemophilia. So you're probably pretty familiar with these 2. This one is a little unusual and not as commonly known, but we just want to have heard about it. These are actually going to be sex-linked. So they are found in a higher prevalence or incidence in males. Because remember, males are X, Y, and so whatever is shown on that X, if it's a little b, that's what's going to be shown in their physical appearance. And so what we need to be able to do is look at a pedigree and then be able to identify if it's autosomal or sex-linked, if it's dominant or recessive. Here, it's actually a little bit misleading as well because we don't see it skip any generations. But feel confident that on the AP exam, they're not going to give you one that is this tricky. They're going to give you one with a pattern that is more easily identifiable. But one of the things that we see is this higher incidence among males. We see that if a woman has it, her son has it. If a daughter has it, her dad has it. So I wouldn't worry very much about this pedigree, but it was the only one for hemophilia that was out there. Red-green color blindness, most people understand the basic idea of this. We're not going to get into the physiology of why you can't tell red from green, as much as know that it has higher incidence as males, it's sex-linked. This is an example of where it's not the brain that's the problem, it's actually the muscle that's the problem. And so there's a lot of muscle weakness here, again, found higher in males. Hemophilia, this one's really commonly talked about. There's a pedigree in your book that looks at the royal bloodline in Russia. The main idea is that your blood doesn't clot, and so you're missing actually a specific protein to help your blood clot. So that's the end of the gene diseases, but it turns out that there are other diseases that you can inherit, and these diseases involve entire chromosomes. So they're still inherited because they're still passed from parent to child. But this time we're not talking about a gene, we're talking about an entire chromosome. Now this is called a karyotype. It is a picture of the chromosomes. And what we should be able to see is that our chromosomes come in pairs. That is the normal state, chromosomes that come in pairs. Now these pictures are sometimes a little misleading because it looks like this one's all messed up. But remember, we're talking about getting a picture of the chromosomes, so it's not that we can tell them to line up perfectly. Also, these are pictures of singled chromosomes. It's much easier to get pictures of doubled chromosomes because they're copied and doubled and lining up very nicely during, for example, cell division. But if we want pictures of single chromosomes, then we actually have to add certain chemicals to the cells to get the DNA to coil up on its own. The main thing that we look for is this concept of being in pairs. So there's 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2, 2. And we get down here to the sex chromosomes, and what we have is an X and then nothing. There's no second X, there's no Y. And this is actually Turner syndrome, XO. And so the main thing that you're going to be asked to do is identify if there is an abnormality, and then you're going to be asked to explain why. Okay? So sometimes a really common one is called trisomy 21. Well, our chromosomes are supposed to come in pairs. Tri actually means 3 of number 21, and that is Down syndrome, not Downs, Down syndrome. And they're going to have very characteristic features. There's almost always mental retardation, specific facial features, usually shorter in stature. And this is because it's not just one gene that's wrong, it's a whole collection of genes, where there's either too much or too many. Now, why does this happen? This happens because of an error in meiosis. If it's passed on from parent to offspring, that means that during meiosis there was some sort of error during anaphase or metaphase, and there were too many chromosomes. And so this is called aneuploidy. Right? We recognize ploid as number of chromosomes. U means true, and an would mean not. So the word aneuploidy literally means not true number. This error is actually called non-disjunction, and it occurs again because there's some sort of error in meiosis. And so either the sperm or the egg has too many or too few. Keep in mind, if there's too many in one egg, there must be too few in the other. Klinefelter is XXY, so this is where there are two X's and a Y. And so this means that there was either a problem in the sperm, and the sperm came with an X and a Y, or it's a problem with the female and the egg gave both of the X's. So the big concept is that there's this huge host of genetic or inherited diseases. Some are problems with just specific genes, and they have very specific patterns to inherit. Others are problems with whole chromosomes. You do want to go ahead and try to learn these three. Downs is trisomy 21. Klinefelter is trisomy of the sex chromosome. And Turner is monosomy, only one of the sex chromosome. There are a lot of other ways that this could go wrong, but most of them aren't viable embryos. So even if the egg is released and fertilized by a sperm, even if it starts dividing, a lot of times that's why women will have miscarriages, because there was actually something wrong with the egg. One of the biggest questions is, if we have advanced technology, we can actually test for this. So very early on, they can remove cells from the mother's uterus, and they can look at the baby's cells and determine if the baby has a chromosome disease. A little bit later, they can remove the baby cells and actually test the genes of the baby. And so the biggest question is this idea of morality and ethics. Should we even be testing? And what should we do with the results of that test? So a big concept is with pregnancies. Would a mother terminate a pregnancy? Would a mother use that information to go to classes and be more prepared? Does it actually benefit the child if the parents know ahead of time? As a parent, would you want to decide whether to get pregnant or not get pregnant if these run in your family? Another moral issue is actually with insurance. So, for example, with Huntington's, everybody could go and get tested for Huntington's, and we would know who would get it later. And so should insurance companies require you to get tested? Should insurance companies have access to your testing? Should they be able to charge you a higher rate if you're at a higher risk? So there are a lot of moral issues that come up with that as well. And that's all.